So, welcome to the tutorial session. So, the first question is about cholesterol. Cholesterol is an important constituent of eukaryotic cell membranes and affects the fluidity of the membrane. So, suggest how it might be oriented in the membrane. So, that is the question is how cholesterol is going to be oriented in the cell membrane. So, in order to address this question, let us first look at the molecule. So, if you look at cholesterol, you can see here that there is a polar hydroxyl group because of the electronegative atom oxygen, and this polar hydroxyl group can also involve itself in hydrogen bonding. So, you can think about electronegative atom being involved in hydrogen bonding. Also, the oxygen of this can also be involved in hydrogen bonding, right. So, these are all possible interactions with this hydroxyl group. If you look at the rest of the molecule is essentially devoid of any polar groups, right, which means that it would be quite hydrophobic. So, if you look at what this end of the molecule would interact with, this end of the molecule would probably interact very well with hydrophobic molecules, and clearly it will repel groups which are highly polar, right. So, even though there is perhaps a little bit of possibility of some pi stacking with this olefin, I think it is one can imagine that it is going to be well buried in the molecule, that it may not be very important. So, if I have to break down this molecule, I would break this down into a large rectangle which is hydrophobic in nature, and a small hydroxyl group which is going to be polar, right. So, let us keep this in mind when we are looking at how it is going to interact with the lipid membrane. The membrane, as we know, consists of a polar head group and a non-polar or hydrophobic tail. So, you can approximate the lipid membrane to be something like this. So, if cholesterol were to interact with this group, what we could predict is that the hydrophobic regions are going to come closer to one another, and the hydrophilic groups are going to be coming close to one another. So, here let us mark out the hydrophobic and hydrophilic regions. So, this is the hydrophilic region, and the rest of the molecule over here is actually hydrophobic. So, with this in mind, if we were to predict how the lipid membrane would interact with cholesterol, then one could draw out a figure like this. So, you have the lipid membrane over here, and as we looked at earlier, cholesterol is basically a structure like this, and so it would interact in the following manner. Perhaps there will be another molecule of cholesterol over here with the OH pointing out here. So, what this would do is that the hydrophilic interactions would be favored over here, and the hydrophobic interactions would be favorable over here. Okay. So, that takes care of this question. So, the next question is compare the hydrogen bond accepting capability of an amine, RNHR and aniline. Let us first draw the structure of the amine. So, here is the structure of the amine. Next, let us draw the structure of aniline. This is the structure of aniline, and here is the lone pair that we are interested in. And the question is about how well it accepts a hydrogen bond. So, now therefore, this lone pair becomes important for us to understand, right, and this is the lone pair that is going to interact with a hydrogen bond, let us say with water. Okay. So, if I have to compare these two, then there are certain characteristics that we need to understand over here. The first thing is that in aniline being since the nitrogen is attached to the aromatic ring, it is quite possible that it is going to interact with the aromatic ring. So, the way this might happen is that you may have some sort of a resonance form that one could draw, where you have a negative charge on inside the aromatic ring. Okay. So, you have a positive charge over here and a negative charge over here. So, because of this, what might happen is that you will have a pull of this electron pair of this lone pair on nitrogen into the benzene ring. This perhaps makes the lone pair less available for hydrogen bond. So, therefore, based on this, one would argue that aniline would have a less hydrogen bond accepting capability compared to RNHR. Next question is, arrange the following functional groups in their order of hydrogen bond accepting capability, which is ester, amide, ketone and carboxylate. So, first let us draw the structures of these. Ester is R, C double bond O, O R. Amide is R, C double bond O, NH2. Ketone is R, C double bond O, R, and carboxylate is R, C double bond O, O minus. Okay. So, based on this, we can now arrange it in the order of hydrogen bond accepting capability. So, the order is that since carboxylate has a full negative charge on it, it is more capable of accepting a hydrogen bond when compared with a ketone or an ester. Okay. So, these are easy to compare, right. So, now let us come to the amide shortly, but the fact that the greater the electron density on the carbonyl oxygen, the stronger it will act as a hydrogen bond acceptor. So, because of this, the carboxylate is clearly the best hydrogen bond acceptor in this series. Now, if you think about the amide is very interesting, because the carbonyl oxygen of the amide will act as a good hydrogen bond acceptor, since the lone pair of electrons of the nitrogen can interact with the carbonyl and increase the electron density of the carbonyl oxygen. So, what ends up happening in the amide is a situation like this, where this lone pair is going to go inside and get delocalized, and actually it is going to form a CO minus, which the CO minus is going to have a slight negative charge on the oxygen. So, consequently, the carbonyl oxygen in this functional group is a much better electron hydrogen bond acceptor compared to a ketone and an ester. So, in the case of a ketone and ester, the electronegative OR is going to pull electrons from the carbonyl and therefore makes the carbonyl a poorer hydrogen bond acceptor. The next question is, what is the typical strength of a van der Waals interaction in kilocalories per mole? Is it stronger than a salt bridge? So, in order to answer this question, let us recap a little bit about what van der Waals interactions are. So, van der Waals interactions are nothing but very weak interactions which are weaker than hydrogen bonds that can take place between two hydrophobic regions of a protein. So, here the example that we looked at is a leucine residue interacting with a valine residue. And so, this type of interaction, where you have two hydrophobic groups coming close to each other, is called as the van der Waals reactions, van der Waals interaction. The bond strength of van der Waals interaction is typically less than 2 kilocalories per mole. So, now if you look at the salt ionic bond or salt bridge, an ionic bond is formed between typically a carboxylate ion of an acidic residue, such as the one shown here in aspartic acid, and an amine from a neighboring residue, such as lysine or histidine. So, here in this example, the protonated lysine has a full positive charge on it, and so this kind of an interaction is called as a salt bridge. The bond strength here is somewhere between estimated to be 5 to 10 kilocalories per mole. So, the answer to the question is the salt bridge is a stronger interaction compared to van der Waals. Okay. Now, let us look at the next question. The PAW protein is an important protein in signaling processes within the cell. It exists freely in the cell cytoplasm, but must become anchored to the inner surface of the cell membrane in order to carry out its function. The question is, what kind of modification to the protein might take place to allow this to happen? So, let us look at this question in a little bit of detail. So, here is this protein, PAW protein, which is freely floating around in the cytoplasm. Now, for it to be activated, it needs to go and bind to the surface of the membrane. So, in order for this to happen, we have already seen that the lipid bilayer is the major component of the cell membrane, and so you can imagine that the lipid bilayer is going to be like this, right? And if it has to associate itself with the lipid bilayer, then it would need some hydrophobic residue to actually associate itself with, right. So, therefore, the protein can have multiple lipid groups covalently attached to it, but where the lipid binds to the protein depends on the lipid group and the protein. Okay. So, once this lipid group is attached to the protein, one of the examples of this is farnesylation, which is a type of prenylation and involves adding an isoprenyl group to the C terminus of the enzyme of the protein. So, once this is added, it creates a hydrophobic surface for it to interact with the membrane. So, it is likely that the protein PAW will have to probably undergo some sort of a modification with a hydrophobic group for it to go and interact with the lipid membrane. Okay. The next question is, most unsaturated alkyl chains in phospholipids are cis rather than trans. Consider the cis unsaturated alkyne chain in the phospholipid, then let us redraw it to give a better representation of its shape and compare this shape with the trans isomer. What are the conclusions that you can make regarding the packing of such chains in the cell membrane? So, if we were to draw a cis unsaturated lipid, it would be in the following manner, where you have a cis double bond over here. Similarly, if you were to draw this in the trans configuration, you would get the structure like this. So, as you can see, the cis configuration actually undergoes a change in the direction, and this is going to increase the or it is going to make the regular packing of this more difficult. Whereas, in the trans you have an orientation like this, which can improve the packing. So, therefore, cis double bond introduces a kink in the cellular chain, which will hinder the regular packing of the hydrophobic chain and increase the fluidity of the cell membrane. The hormone adrenaline interacts with proteins located on the surface of the cell and does not cross the membrane. So, adrenaline structure is shown here. However, larger steroid molecules such as estrone cross the cell membrane and interact with the proteins located inside the cell nucleus. Why is a large steroid molecule able to cross the cell membrane when a smaller molecule such as adrenaline cannot? So, in order to address this question, let us look at the structures of these molecules once again. So, adrenaline, as shown here, is more likely to be protonated, and it would have a structure like this, where you have two hydroxyl groups and you have this aliphatic hydroxyl group, and you have NH, Me, perhaps this is going to be protonated, and you will have a positively charged species. So, as we looked at earlier, in a cell the lipid bilayer is present, and this is going to be hydrophobic. hydrophobic. Okay. So, in order for the compound to get in, it has to get in to the hydrophobic region and then get out of this into the cell. So, considering the positive charge that is present over here, one would predict or one can understand why adrenaline does not get into the cell. So, we have already looked at this previously. One of the ways in which nature solves this is to create small areas wherein such small molecules can get in, small polar molecules can get in. So, these are transporters which help with taking in this highly polar small molecules. Whereas, estrone having a hydrophobic, highly hydrophobic surface can actually better interact with the membrane, it can get in and then it can also get across the membrane because it dissolves or it can interact, it can there can be a favorable interaction with the lipid membrane which can allow it to permeate. So, which is why estrone can get in and then go all the way to the nucleus and exert its effect. Next question is, explain why the lock and key hypothesis does not explain enzyme function. Okay. So, in order to understand this, first look at let us look at what lock and key hypothesis is. Lock and key hypothesis is nothing but the enzyme has a surface or has an area where the substrate, which is the key, will come and bind to it. And since the lock and the key have exactly very well paired shapes, the key can go in and interact with the lock and maybe there it binds and then it is going to turn over. However, this scenario does not explain how some enzymes can catalyze a reaction on a range of different substrates. So, the lock and key hypothesis would necessarily or would tell us that the substrate shape and size has to be very highly conserved, but that is not the case in certain enzymes. Some enzymes bind and react with a number of substrates, and so this lock and key hypothesis, while it is useful, does not explain all the functions of an enzyme. So, because this in turn would imply that the catalyzed reaction is only efficient for the optimum substrate. As this is not the case for many enzymes, the lock and key hypothesis must be invalid. So, instead, the hypothesis that explains a number of the observations better is the induced fit hypothesis. So, here the binding of the substrate forces the active site to change shape when it enters. It is like some sort of a molding process that happens, and once that happens, you can have the substrate fit into the pocket much better, right. So, enzyme changes shape such that the substrate can fit, and the substrate can also change its shape a little bit to accommodate itself into the enzyme. So, here is the example that we looked at previously, where you have a carboxylate ion interacting with an ammonium through ionic bridge, and this carbonyl interacting with through hydrogen bond with the substrate. Now, after it binds, it is possible that this van der Waals interaction, which was previously quite weak, can now come and wrap itself around, and so that is the conformation change that is required for the induced fit to occur. So, how does this explain the diversity of substrates? So, each substrate induces the active site into a shape that is ideal for it, and so even with a different substrate, one can have the same effect, right, or the same binding or similar binding, because this is a molding process, and this molding process does not distort the active site, right. So, the reaction mechanism happens quite consistently even if the substrate is different. Also, the substrate is not a passive spectator, as we have looked at previously, and it also changes shape to accommodate itself into the active site. So, as the enzyme changes shape to maximize bonding interactions, the same thing can also happen to the substrate. So, therefore, this model allows us to explain why there is a large number of substrates that can be turned over by a particular enzyme, even though the substrate may look very different. It is quite likely that the interactions of the active site with the substrate is quite similar, and therefore, it can recognize substrates which are similar in not just in shape, but also in binding interactions. next question is, why are transport proteins essential for normal functioning of the cell? So, we have already seen this, the structure of the lipid bilayer is shown below, and now polar molecules, as we discussed earlier, cannot enter because they do not get into the hydrophobic surface. So, in order for this to happen, we have looked at in the previous case of adrenaline, the transport proteins can act as carriers. So, the way in which we understand these transport proteins is that when there is a polar charged molecule, it goes and binds itself to the protein surface, the transport protein surface, and then the protein acts as if it is a carrier. It wraps itself around the polar molecule, and then through a process of diffusion, the molecule can get out. Okay. So, this is very important when neurotransmitters have to be transported back into the neuron, so that they have a limited period of activity. So, the transport proteins float freely within the cell membrane because they have hydrophobic residues on their outer surface, which interact favorably with the center of the cell membrane. The portion of the transport protein that is exposed to the surface contains a binding site that can bind the polar molecule. So, therefore, transport proteins are very important for the functioning of the cell. So, they act as something like they stow it away, hydrophilic pocket, ferry it across the membrane and release it on the other side. Okay. Therefore, transport proteins are very important for the normal functioning of a cell. The next question is, what is unique about glycine when compared with other naturally occurring amino acids? So, let us draw out the structure of glycine. Glycine is NH2 COOH. Okay. So, one thing that really stands out here is that this is an achiral molecule. So, this is one thing that is very different unique about glycine when compared with other naturally occurring amino acids. If you go and look at the structure of all the other amino acids, they will have a chiral center in it. Next question is, draw out an energy profile to show how enzymes catalyze reactions. Okay. So, in order to do this task, let us first draw out the coordinates, which we have looked at several times. So, here is your reaction coordinate, and here is energy. It can be free energy or delta G, for example, and so on. So, let us draw a hypothetical reaction A going to B. Let us assume that it is a exothermic reaction or exergonic reaction. This would be our delta G. Now, what we understand from kinetics is that there is going to be a barrier to this reaction, and this barrier is shown here, which is delta G double dagger. Okay. Now, in order for us to understand how an enzyme catalyzes the reaction, we have already looked at what delta G double dagger is, which I am just redrawing here for convenience. So, if the reaction goes faster, we know that there is going to be either a change in the ground state energy or a change in the activation energy. So, that means that the ground state energy is going to go up, which will result in the substrate being destabilized, so that it can cross the barrier with, let us assume that it is a similar barrier. The other option is that you can have the transition state coming down in energy, and what may end up happening is, if you have a combination of these two, the enzyme is going to go much faster, and it's going to, or the reaction is going to go much faster, and the turnover is going to be extremely fast. So, in reality, it is more likely that an enzyme catalysis may happen in phases such as this. So, here is the starting material A, here is the product B, this is the energy, and this is the reaction coordinate. Okay. Now, Next question is, consider the structures of adenylin, estrone and cholesterol and suggest the kind of interactions that are possible in these molecules. And the interactions that we are interested in is intermolecular interactions. So, we have already looked at adenylin previously. So, adenylin molecule is over here. So, it can undergo protonation and form NH2 Me plus, that will create a possibility of a salt bridge type interaction. You also have a hydrogen bonding capability over here, and this will give us a pi interaction over here. In the case of estrone, there is a hydrogen bonding capability here, pi stacking over here, there is a hydrogen bonding capability here, and the rest of the molecule is actually hydrophobic. Cholesterol we have already looked at previously. It is going to have a large hydrophobic interaction and a small area where it can do polar interactions. So, the answer is drawn out here. So, you can see here that there are hydrogen bond donors, there are hydrogen bond acceptors in the case of adenylin, and you have a hydrogen bond donor over here, hydrogen bond acceptor over here, and here is another hydrogen bond acceptor donor pair. And as we discussed earlier, if it is ionized, then if it is protonated, then it is going to be ionic over here, and the rest of the hydrogen bond capability is similar. In the case of cholesterol, there is some possibility of van der Waals interaction, and there is a hydrogen bond acceptor donor pair over here. Similarly, in the case of estrone, is going to be like this, and there is also a possibility of hydrogen bond acceptance and a dipole-dipole interaction. So, now let us look at a situation where a protein has the following cavity in it. Identify the major interactions that occur. So, if you see here, the major interactions that might occur here are, here would be an ionic interaction or salt bridge interaction, right. Here would be a hydrophobic interaction, here would be a pi stacking, van der Waals, right. And here is a situation where you would have hydrogen bonding occurring through this hydroxyl group. The next question is, the tertiary structure of many enzymes is significantly altered by phosphorylation of serine, threonine or tyrosine. Identify the functional groups that are involved in these phosphorylations and suggest why these phosphorylation might affect the tertiary structure. In order to answer this question, let us first look at what phosphorylation is. Phosphorylation is a situation where you have an alcohol in the form of a serine or threonine or tyrosine, and it undergoes a phosphorylation which involves addition of a phosphate group on it, and what was previously a situation where you had only a hydrogen bond acceptor and a hydrogen bond donor now is converted to a functional group which is ionic in nature. So, what happens in this process is that you have altered the polarity of the residue. Okay. So, you have converted a simple little relatively polar group into an ionic group. Okay. So, this is an example of a post-translational modification that we have looked at previously. And post-translational modification, translation is the process by which a protein is synthesized in the cell, and many proteins are altered after they are translated. Okay. So, for example, N termini of many proteins are acetylated, making these proteins more resistant to degradation. And we will also look at later that acetylation of proteins has a major role to play in the control of transcription. Okay. Now, once we do phosphorylation, the serine, threonine or tyrosine residues are, once they are phosphorylated, they can be a part of many signaling pathways, and therefore, it is a very important post-translational modification that occurs. But now, because of the strong negative charge on the phosphate group, this completely changes the way in which the protein is shaped and how it interacts with water. So, a protein that normally does not interact with water will suddenly become hydrophilic and become water friendly when it is phosphorylated. So, this change results in the major biochemical properties of the protein, and so therefore, it has been used, phosphorylation has been used to carry out many signaling events inside the cell. The next question is, lactate dehydrogenase has a 1000-fold selectivity for lactate as a substrate over malate. However, if a mutation occurs that alters an active site glutamine residue to an arginine residue, the enzyme shows a 10,000-fold increase in selectivity. Okay. So, explain this astonishing transformation. So, here are the structures of lactate and malate. So, if you look at these two structures closely, the major difference is here a methyl group which is hydrophobic is converted to a carboxylate which is an ionic residue. So, ionic bonding. So, let us first look at the reaction. Here is the molecule pyruvic acid. It interacts with lactate dehydrogenase and gives you lactic acid as the product. Okay. So, here the possible interactions that we have looked at in the past are the ionic interaction, which is shown here, hydrogen bonding interaction and hydrophobic interactions or van der Waals interactions. So, ionic bonding plays a relatively minor role in the tertiary structure compared with hydrogen bonding or van der Waals interaction, but it can play a crucial role when it is binding to the substrate of an active site. So, here it is redrawn, and one can imagine that the active site of the protein must contain an area where it binds to this carbonyl group. It must have an ionic bond over here and a hydrophobic or a van der Waals interaction. Okay. Our data tells us that the reaction of this is much faster when compared to So, therefore, it is possible that the arginine binds better and stabilizes the transition state better in the case of malate when compared to the lactate. So, this is structures of lactate and malate as shown here, and when we mutate the glutamine residue to the arginine residue is quite likely to bind to the carboxylate much better when compared to glutamine. So, from this example, what is clear is that by changing the residue in the protein's active site, or it is possible that the substrate specificity can be altered. So, here we can the enzyme has become more selective towards malate when compared to lactate. Okay. Next question is, the chains of several cell membrane bound proteins wind back and forth through the cell membrane such that some parts of the protein structure are extracellular and some parts of the protein are intracellular. Okay. So, the question here is, how might the primary structure of a protein help in distinguishing the portions of the protein embedded within the cell membrane from those that are not. Okay. So, I have drawn here a cartoon which shows a protein which goes back and forth into the membrane and out. So, here you have an extracellular domain over here, and then there is a region where it goes inside or embeds itself into the membrane, and then there is another loop, and then again it goes through the membrane, then it goes out, comes back in, goes in, comes out, and so on. So, there are several loops here going back and forth. So, we already know that if a sequence of amino acids with hydrophobic side chains are identified in the primary structure, that this hydrophobic side chains will most likely interact with the lipids in the lipid bilayer. So, it is possible that this is the transmembrane region, okay, because they are located within the cell membrane as shown. This is because hydrophobic side chains would interact more favorably with the fatty cell membrane than the aqueous environment on the either side of the cell membrane. So, regions that are of the protein that are predominantly made of polar side chains are almost certainly located intracellularly or extracellularly. What we could understand here is that the regions which are over here are likely to be hydrophobic, and the regions which are over here are going to be most likely polar. Okay, so you have again binding regions and so on, right. So these are some predictions that one could make based on our understanding of how the cell membrane works and the hydrophobicity and hydrophilicity. However, it would be wrong to conclude that all amino acids with hydrophobic side chains are located in the transmembrane region, and all amino acids with polar side chains are located intracellularly or extracellularly, because it is possible that they are particular shape wherein the polarity becomes less important or more important.